All right. Welcome. Today, we're going to put on our detective hats and head into the micro lab to solve a classic whodunit. Our mission? To unmask a bacterial culprit, Enterobacter cloacae, by following a trail of biochemical clues. Let's get right into it.
So imagine this. You're the lead detective. A mystery sample lands on your bench. The first report comes back. It's a gram-negative rod. Now, this is our starting point for sure, but the list of potential suspects is, well, it's long. So how do we even begin our investigation?
Every good investigation starts by looking at the initial evidence. And for us, that means we're kicking things off with the most fundamental test in all of microbiology, the gram stain.
And here it is. Under the microscope, we see these really distinct pink rod-shaped bacteria. So that confirms they're gram-negative. It's a fantastic first clue. It really is. But here's the problem. Some of our most common suspects, I'm talking E. coli, Klebsiella, they look exactly like this. So yeah, we're on the right track, but we're going to need a lot more evidence. Okay, so to start narrowing down this suspect list, we need to see how our organism behaves. We're going to grow it on a special type of culture medium. You can think of it like we're laying a trap, trying to get it to reveal a little more about its true identity.
Now, this is where things get really interesting. This is the result on what's called Mac-Conkey agar. See those super bright pink colonies? That color tells us our culprit is a lactose fermenter. This is a huge step forward. But it also kind of deepens the mystery, because another one of our major suspects, Klebsiella, is also famous for fermenting lactose. So the plot thickens.
All right, let's get our lineup straight. Based on these first few clues, we're looking at three main suspects: E. coli, Klebsiella, and our primary target, Enterobacter. To really tell them apart, we're going to have to dig a whole lot deeper.
Okay, now for the really cool part. We're moving on to a series of biochemical tests. You should really think of this as a direct interrogation. We're going to force the bacteria into a bunch of different chemical situations just to see how they react. We're making them reveal their unique metabolic fingerprint.
So, our first interrogation tool is the triple sugar iron test, or TSI for short. As you can see right here, this one little tube is just packed with clues. It's got 3 different sugars: glucose, lactose, and sucrose, and a pH indicator that'll change color and tell us if the bacteria are fermenting them.
And the result is yellow over yellow, which we call A/A.
And you can see some gas production in there too. So this shows us that our organism is a really powerful fermenter, just munching away on multiple sugars. But here's the twist. E. coli and Klebsiella give the exact same result, so we still can't tell them apart. We're stuck.
We're at a really critical point in this whole investigation. We need a test that's finally going to break these suspects apart. So, what is it? What's the game-changing clue that we've been looking for this whole time?
And boom, here it is, motility. In this test, we literally just stabbed the bacteria into a semi-solid medium. On the left, you can see how our suspect has swam all over the place, spreading out from that stab line and making the whole tube cloudy. That means it's motile. Now compare that to the tube on the right, where the bacteria stay put right where we left them. This is the key difference that lets us rule out Klebsiella, which is famously non-motile. One suspect down.
Okay, next up, we're going to confirm our hunch with the citrate test. Basically, we're asking, can this organism use citrate as its only food source? And the answer is a resounding yes. Look at that. It turns the medium from its original green to this brilliant royal blue. This is another massive piece of evidence, because our other remaining suspect, E. coli, is typically citrate negative.
So, we've gathered all our evidence. We've got the gram stain, the culture plates, the results from our biochemical interrogation. Now it's time. Time to put it all together and make our final identification.
Let's take a look at the final lineup. And seriously, this table right here is absolute exam gold, so listen up. E. coli is motile, but it's citrate negative. Klebsiella is citrate positive, but it's non-motile. There's only one suspect that fits our evidence profile perfectly, motile and citrate positive, and that, of course, is Enterobacter. And there you have it. That's the key formula to lock away in your brain. Motility positive plus citrate positive equals Enterobacter. Case closed.
So now that we know who it is, let's take a look at what it does. Every culprit has a modus operandi, an MO. And for bacteria, that really means its clinical behavior and how it impacts the human body.
So if we look at its rap sheet, Enterobacter is a major player in what we call nosocomial, or hospital-acquired infections. It's a real opportunist, often causing things like urinary tract infections, pneumonia,
and even bloodstream infections, especially in patients who are already vulnerable.
Okay, now this is a really, really important clinical concept. You've got to know this. Enterobacter carries a gene for an enzyme called AMP-C beta-lactamase. What that means is it has this nasty trick up its sleeve. It can actually become resistant to certain antibiotics while a patient is being treated. I mean, think about that. It's a high-yield topic for a reason and a huge challenge for doctors.
So how does our body even fight back against this thing? Well, Enterobacter's outer membrane has something on it called LPS, or endotoxin, and it acts like a giant red flag. It triggers this powerful inflammatory alarm which calls in the immune system's first responders, the neutrophils, to come in and attack the invasion.
We've gone from the crime scene to the interrogation to the clinical MO. So now it's time for you to put your newfound detective skills to the test. Okay, here's a classic exam-style question for you. You've isolated a gram-negative rod that ferments lactose. Your key biochemical tests show that it is motile and it's citrate positive. Based on everything we've just covered, what is the organism?
If you said Enterobacter, you are absolutely correct. You've cracked the case. By following those clues one by one, we can confidently pick this important pathogen out of a lineup. Fantastic work, detective.